Hello, this is your host Todd Lewis, the Praise of Fly podcast. Don't forget to like, share, and subscribe, and hit the notification bell. Don't forget to follow me on Telegram, Twitter, and Facebook, and support me on Subscribestar, Locals, and Buy Me a Coffee. I am once again joined by Florian. Glad to have you here, Florian.
Hello, glad to be here.
We are here to talk about the revolutionary discovery in paleontology of the soft tissues found in dinosaurs. Now, the original discovery was published by Mary Schweitzer in 2005. I think the original discovery actually was made in 1990, but as she states, she sat on it a while because she didn't want the blowback from the paleontological community. Because what happens, the definition of a fossil is a bone that's been preserved where all the organic material has been mineralized. So if you were to dissolve it in acid, you wouldn't actually expect to find anything. In fact, nobody would have thought you could have had any soft tissue of any kind whatsoever from an animal that old. I think the specimen she used was a 68 million year old Tyrannosaurus rex. Now, other discoveries have included in 2009, Schweitzer and her colleagues found some in a duck-billed hadrosaur, dated to 80 million years. Then there was an Edmontosaurus from 2007. A Triceratops from Montana. This was a follow up, not by Schweitzer, but by other individuals. A Silurisaur in 1998. Possibly a Gorgosaurus, which was in 2015, and then a Lifungosaurus in 2017. And then there's so we have a whole gamut of dinosaurs that have had this various soft tissues, including blood vessels, bone cells, collagen, and other sorts of proteins like hemoglobin. And so this was a huge bombshell. And famously, when she reported it to Jack Horner, he said, "The creationists are going to love you, Mary." And then she says, "Well, it's your damn fossil, Jack." So they kind of knew some of the implications already. Now you have to remember, though, this team is going to get it from both ends. The creationists are going to take that information and use it, and the scientific establishment is going to come down hard on this. The initial claim was that it was contamination, that whichever she found had been contaminated. And I think I asked Grok, I think around 2015, the consensus built up around that okay, it's not contamination. And so before the show, though, Florian, you looked at her study, and you have some slides ready for us. So why don't you make that presentation?
All right, yeah. Can you hear me? Am I
Yeah, we can hear you just fine,
okay?
Florian.
Perfect. Yeah. So just to recall for everyone what kind of dinosaur we're looking at. It's a Tyrannosaurus rex. It's this dinosaur here, or dinosaur skeleton. And specifically, what we're looking at is this bone here, which is called the femur. It's everyone has it. We also have one, and it's basically our upper leg bone, right? And this bone is fossilized, and Schweitzer and her team looked at that. And it's useful maybe to just have a quick look at what constitutes a bone. A bone will have an area of growth here. It's called a spongy bone, or I mean, there are many bones, but we're looking at the elongated bone, like the femur. And then there's bone marrow here. There's some blood vessels going through it. And there's this epiphyseal plate that separates the growth area from the stable area here. And so there's a cortex here, and then there's the inside, which is hollow, and there's bone marrow inside, right? It's called a medullary cavity. And when we take a cross section along a line like this, perpendicular to the screen, we can see several of these canals. So-called osteons. Those are the those are the basic units of bone, basically. And there's a lot of different cells here, and something so-called canaliculi, haversian canals, and then there's other canals, Volkmann canals, that are perpendicular to it. And it's basically, a bone is not just a bone. It's like there's like a lot of nutrients going in and out, oxygen, carbon dioxide. There's blood vessels and nerves going through the bone, and most of them going through these canals, right? And I'm mentioning that because that some of the fossil structures Schweitzer mentioned in the paper are bringing these canals up. So what she found first was when they used acid or used chemicals to dissolve away the fossilized bone, the minerals, they were left with some soft tissue, surprisingly, and these are actually not just some fragment of like tissue or whatever. Those are actually pliable, elastic, soft tissues here. So basically, you can tear it along this little bridge here, and it falls back into shape. And it's also important to notify it's zero point 0.5 millimeter here. So what we're looking is like a 2 to 3 millimeter long speck of material here. So it's not like there's an entire blood vessel in large scale. It's like some small area here. It's basically like a little dust pack, and they magnified it with a microscope here. And when they take the bone away, the fossil material away, you have even these elastic stuff here. So it looks like real, like a slightly degraded tissue. And when they found in tissue in the past, they found some tissue associated with fossils, but that tissue was pretty much dead. This tissue here is like pliable, elastic, and I don't want to say alive, but it's kind of like really biologic. You got that impression, or Schweitzer got that impression. And then they looked a little bit further into what the structure was. When they demineralized the bone, like dissolve all the material away, they are left with transparent structures look like blood vessels. Like here, you see like a blood vessel that's branching here. That's what they found. And the little F here are fenesters, like windows where these canals are intersecting, and so it's a functioning blood vessel system that they isolated basically from those dinosaurs, from this Tyrannosaurus Rex. Here's another part. There's a little biological gunk here, and then they have this network out of blood vessels. Now, one important thing to note is that this here was taken from the inside of the medullary cavity, right? So here from the inside of that wall, and so you can imagine it's probably just very well conserved or very well preserved inside the bone, right? There was no outside weather that was eroding it or anything. It was nice and smug inside the bone. For some reason, though, when they then looked at the canals, that was from the cortex of the bone. So there's one strange thing in their paper, which is basically it's not always the same area of the bone we're looking at. And I don't know whether maybe that's how they do it in archaeology, but that was something that was that I noticed. And here they had to make some kind of sense out of this network, and they speculated blood vessels, and then they used to use a similar structure. They took a bone from an ostrich, like extant bird that's still there, but evolutionary related to dinosaurs, and they dissolved that bone from an ostrich in acid as well, and they found similar structures like these blood vessels also being isolated here, right? So these are probably blood vessels. They also looked at, they don't have pictures of that, but they also looked whether it's just plant material or fungi forming some kind of like meshwork, and it was negative in both cases, right? These images show a little bit more close-up views on these blood vessels. You see some cells here that look like blood cells, erythrocytes, red blood cells, which is also something that she found in there, right? And then they zoom further into it. So they use scanning electron microscopy, and in this close-up, you have this blood vessel structure, and this is the dinosaur here. This is the ostrich here. So these things really look like ostrich structures. They could like really like blood vessels here, right? And they show that it's a blood vessel by showing that the ostrich has very similar structures here. So this is probably a blood vessel, and they found some additional points and some kind of like a profile inside these blood vessels. And so it looks like pretty much they're looking at actual blood vessels. Here could be some blood cells in there. Here these little circles. And yeah, so they're really convinced that it's blood vessels what they're looking at, and then they go more in detail, and now they show other things that look like actual cells. You see, these are cells on the scanning electron microscopy, and they have these small filopodia like appendages, right? So these are small appendages that help a cell when it wants to move, to feel the underground or feel the substrate on which the cell is moving. The cells use these filopodia, these little hair-like appendages, to make contact with other cells. So this is pretty much a cell. It's difficult to imagine how any other structure but a cell would have these little appendages here. And once again, with ostriches, they have similar appendages here. So I'm sure it was very exciting for the researchers around Mary Schweitzer and Jack Horner. And so then they have some supplemental pictures here. They have another version of the defossilized mineral. You can see here like some kind of blood vessels with blackened blood cells here in there. Another view is it becomes very transparent when you dissolve it in acid, and then you still have these little blood cells here. They are black here, and here they are red. It's a little bit different ways to use microscopy to look at those. So if you look at them directly, I think they would appear red. In that image, they just appear black, but they're the same red blood cells that you see here. And then they have another close-up here on the surface of these blood vessels. Let's see another blood cells here. They really look like red blood vessels here, right? And then they look at this is from ostrich and from chicken. So really, these structures look similar to actual red blood cells. So that's one point where they show that this is really red blood cells, or where they speculate that this is really red blood cells. This is supplementary picture, and the story with supplementary pictures in papers is usually they send you send a manuscript in, and then there's peer review, and the peer reviewers demand that you have some additional experiments, and those additional experiments are usually put into supplementary material. So this might as well something that the reviewers have demanded Mary Schweitzer to deliver before they allowed publication of this paper, right? Here they have another examples of blood vessels, and the specialty of this is that this is from another T-Rex specimen from the same excavation site. I think it's in Montana, and so they show that what they have is not just from one dinosaur skeleton, but they have several dinosaur skeleton where they see similar things happening. Blood vessels here, like some growing blood vessels here, also like these little holes. Those are blood vessels cross section, basically. Here you have blood cells again.
Florian, hold on, Florian, we just lost your voice. Florian, we can't hear you. We can't hear you, Florian. Florian. I don't know what to do, guys. I tried to get Florian. I guess we'll just have to wait till he gets out of his system. I messaged him. We'll see what happens. Florian's on a roll, guys. I don't know what to do. Oh man, okay. The slides are almost over. Oh yeah. Well, do we just disconnect Florian and see what happens? Awkward. It's a little awkward, guys. Hopefully he'll realize it soon. I mean, oh, Florian. Yeah, Neurocraft. I have tried to wave him down. I've tried to do all sorts of things, messaging him. Nothing's getting through right now. Florian is yeah, Neurocraft. I'm using StreamYard. I messaged him all the ways I can. So I don't know what to do. Florian, we can't hear you. We can't hear you, Florian.
Yeah, and that's it. That's the paper.
Florian, we couldn't hear you for a long time.
Oh no! What did you miss?
I think it was like the last 4 panels. You went quiet. I tried to get your attention. I messaged you. I waved my hands.
Okay, I'm sorry about that. I looked out for that in the beginning, and it looked like fine. There was no audio issues in the beginning, and so I thought like, okay. So basically, the last 4 panels, you saw them, so I don't need to show them again. But basically, what they found, the basically last 4 panels was they didn't just look at the structures looked like blood vessel. They made actual tests on them, right? So they used antibodies to check for biological residue like collagen and collagen and other proteins that you find inside bone, and they found them there, and then they did a mass spectrometric analysis of the entire atoms they find in these residue, and there's a lot of carbon atoms there. So basically what they say is, look, it looks like blood vessels, and our chemical experiments tell us that it is actually carbon-based material that we find.
Okay, so biological, non-mineral material.
Exactly.
All right. So before we go on, a very generous donor, Mark Honan, for CA $50. Thank you very much. My favorite stream series on YouTube right now. God bless and keep it up, boys. Well, thank you very much. So yeah, there's a lot of things here. The first thing that I think needs to be discussed is nobody. Okay, so prior to Mary Schweischer's team, nobody would have thought that you could find soft tissue in fossils. In fact, I today listened to a podcast interview with Mary Schweischer, and she said the same thing. She said, and she gave the definition of a fossil that I gave you a month or 2 ago, Florian. Where all the biological material has been replaced by minerals. And she says that, well, of course, with her discovery now, that's not. Well, see, here's the thing. If that's the definition of a fossil, why wouldn't a paleontologist just say that's not a fossil then? I mean, Isn't that, I mean, why is that not the answer that was chosen? Because if you do find soft tissue, then by that definition, it's not a fossil, it's something else. Now, you have one of two choices. You could redefine the definition of a fossil, or you could just say it's not a fossil. The problem is if you redefine the definition of a fossil, this looks an awful lot like the redefinition of a vaccine that we all remember just a few years ago.
Yeah. So basically, what they might actually be looking at are dinosaurs that are not really that old, right? And the way they determine that those dinosaurs are 68 million years old, and I think that's also important that people are aware of that. They basically there's no so if something like Ötzi, right? That's like the Stone Age man that they found frozen and mummified in I think in the Austrian Alps or Switzerland Alps. Anyway, this guy is 5,700 years old or something like that, and they can use carbon dating for that because carbon decays in 10,000 years, and no, wrong, carbon decays in 5,730 years. So after 5,000 years, 50% of the radioactive carbon is there, and then after another 5,000 years, 25% is there, and so on and so forth. But carbon decays faster than a million years. If you want to know something that's a million years old, you need to carbon date, or you need to date, not carbon, you need to date with radioactive material from the stone, from the sediment itself. So in those 68 million years old, that is, then we know that it's that old because we can say the stones surrounding those bones, they are actually that old. But then the bones themselves, you cannot show that they are 68 million years old, right? We just say they are. I mean, how else would they gotten into the sediment and be included in the sediment, or surrounded by the sediment, and then they're being preserved in there, and that's how we know fossils. And so if the stones in the sediment are 68 million years old, then the fossils or the animals that were in there once will also be that old.
Yes. Now, and as you said, they would use radiocarbon dating, but the discoverer of the method, Willard Libby, admitted that the upper ceiling is 50,000 years. And this initially was used for archaeology, like human remains, artifacts, it digs, and so on. Now, the other thing then is with this, there's a certain assumptions about the decay rate, which assume that it's a closed system. But that just can't be the case. A closed system is a scientific laboratory experiment. This is in nature. Why would you assume? Why would you assume that the daughter element particles are not being added to or taken away? It's in nature. It's open. It's not. See, if you say that nature is a closed system, in this particular context, you've just destroyed any distinction between a laboratory setting and a nature setting, which you have to have if you're doing science. You can't collapse the two into the same definition because they're not. That to me seems suspicious. But the other thing is with that we have a dating issue, right? Because if we want to date the rocks, using it to get anywhere, 68 million years is what they said was the date for the fossil that Schweitzer was using. But then we find these soft tissues, which now, as far as preservation, when I asked Grok about this earlier, the most shared, so red blood cells. It says here, where was it? I have a few things here. Okay, so they say, so Grok is giving us the upper limit. 50,000 years for collagen, 500,000 years for collagen, and then 100,000 years for red blood cells. And that would be with the current models. Neither one of those are remotely applicable to this. Now, this does create a dating crisis, though, because how can you have this soft tissue when the rock, the fossils in, is dated to be so much older? Well, you don't. Yeah, I mean, you just don't talk about it, right? Because I remember when I first told you about this, you hadn't heard about this, and there's a good reason, because this creates a crisis not only for the age of the fossil, but for accepted dating methods. Because if it really were that old, you wouldn't expect to see this. And in fact, Mary herself says you wouldn't expect to see this. Now, of course, you wouldn't dissolve a fossil in acid if you didn't expect to find something. So the question that I would want to know, and I don't think Mary or Jack have answered this, is what did you expect to find? Because that sounds like to me that they already had doubts about certain aspects of the scientific model. Now, I don't know what those are because they haven't told us, but you don't do this without some goal in mind. And the reason why I say this is in 2006, radio host Bob Einhart asked Jack Horner, he said, if I give you $20,000 for your team, would you carbon 14 date the Tyrannosaurus Rex fossil? And Jack's hemming and hawing, saying, oh, well, you know, one, we're just not going to find carbon 14, so why would you ask us to date it? And two, whatever answer we get would be gibberish. And in the interview, Bob Einhart says, yeah, but if you dissolve a T-Rex bone in acid, you don't expect to find a red blood cell or collagen either. And so here's the problem, right? Jack then later says, this was a creationist ploy, so I'm not going to test carbon 14 on the fossil. Right, but then why would you dissolve the bone in acid? You're not going to find anything. If you're going to default to the standard paradigm, you wouldn't dissolve a bone in acid unless you wanted to find something.
I mean, if you think that's a creationist ploy, then doing the carbon dating would actually completely devalue the creationist ploy.
Yes.
So perfect opportunity to do it, right? So
well, right. I mean, there's no reason not to do it, right? Because
exactly.
One, you get $20,000. You get $20,000 as a grant, so to speak, and then you get to show up the creationists by saying, "Look at these silly creationists." But the fact that he claims to know you're not going to have carbon 14 on the fossil without dating it, one is very unprofessional. I mean, a scientist can't go around saying, "Well, I know for a fact that substance isn't there, so I'm not going to test for it." You don't. I mean, I guess you do get to say that, but it's not very scientific of you to say that.
I mean, do they know that it was they looked at some other dinosaur. I mean, do they know it's from a T-Rex? Maybe it's just a bone found, and it could be just a very some of the dinosaurs survived, right? Some of them, like the smaller versions, they survived the apocalypse if you believe in the planet, the comet version, right? So what if that's a femur of a smaller specimen? Although they mentioned at one point that the femur they're looking at is like 1 meter in size, and that seems like, and the dinosaur, they say that dinosaur, they determined the dinosaur to be 18 years old when it died. I don't know how they do it, but they were very clear it's an 18-year-old roughly dinosaur, and the femur was 1 meter, so it's basically this size. Yeah, that seems like a smaller T-Rex to me. So yeah, maybe it's something else and not really a T-Rex, but let's assume it is. Then either it's kind of sensational because either the T-Rex isn't just that old, or they're looking at artifacts.
Well, right, but you see, here's the problem, right? If it's not just a problem for the fossil itself, right? It's a fossil. There's three problems. There's the first problem is the age of the fossil itself. Okay, that's the first problem. Second problem is the question of dating. So, i.e., the radiocarbon dating of the rock around the fossil. But then this bombshell here. But there's a third problem. It completely upsets the history of Earth. So, if this dinosaur isn't that old, if it's not in fact 65 million years old, and I think you said the upper limit might be 1 million, right, Florian? Was that the upper limit?
The upper limit of what?
Well, the preservation of the red blood cells, the collagen, the soft tissue.
We had the number 1 million. We are using that, but I don't think even proteins keep intact that long. But correct me if I'm wrong. It's not 65 million years or 68 million years.
No, okay. So I was asking about your number. When I asked Grok, the red blood cell upper limit was 100,000 years.
Yeah.
And the collagen, and this would be like remains even, would be 500,000 years. So, but if you're going to have an intact red blood cell, that's 100,000 years. That was what Grok said as the upper limit we know from the calculations. I mean, if that's the case, then this dinosaur is not from the Mesozoic. He would be from the Cenozoic, which would just completely shatter the. Actually, it's a female T Rex. She's from that time. That would shatter the whole history of life if
I
that's the
mean,
case.
it doesn't really. Yeah, it's true, but it doesn't really. It wouldn't even be a. It wouldn't be a complete catastrophic shattering because, like, someone in the comments, like Imperator Forge says, there's depiction of fossils in old Chinese and Greek vases, and I would think they're called mythological features. Yeah, mythology comes from somewhere, right? So um, I mean, do if you want to assume that evolution took place, do dinosaurs have to die for for other animals evolving from
Well,
them?
no,
No, not
well,
really. No,
well,
I mean it could
I
be
guess
that.
it depends, right, Florian? Because growing up, right, and if you when I watched all the dinosaur programs that were available, and even today, the general theory is that the rise of mammals could only occur because dinosaurs went extinct.
Why though? Why would that be?
Well, the arguments that I've heard is that they were at the time, so 65 million years ago, mammals were just the size of like mice. And so there was this downward pressure of competition from the dinosaurs that were large. So consider the first mover advantage, right? Dinosaurs had the first mover advantage. So in order for mammals to break free, they had to be removed, just like old growth forests would have to have the trees removed for the smaller ones to grow up beneath them.
Yeah, that's true, but it could be that just dinosaurs, they withered away in some areas in the world faster than others. So yeah, there were biosystems, habitats where dinosaurs were still alive. Like let's say 50,000 years or something like that. If there were pockets of Earth that still had dinosaurs in them, everywhere else, these creatures were slowly withering away. Then yeah, then you have enough options for mammals to arrive because there's just not as many of the dinosaurs there. In
Well,
fact,
okay,
I mean, if you think if you think the comet didn't spell the end of the dinosaurs, then yeah, it's completely possible to think of ways in which they kind of like hung around and then slowly vanished from the world because their ability to thrive being such huge creatures is probably not going to be forever because it has certain disadvantages to be like a humongous animal like a dinosaur.
well, right. So again, the other okay. So there's actually another problem as well. I'll get to that in a moment. But back to your point. Okay, it's not a problem that quote can't be solved unquote, but it would be a huge earthquake in the understanding of the development of life on the current model if you make that change. But the other problem is, given the upper limit for red blood cells of 100,000 years, uh, this there's going to be a hard limit for dinosaurs, and that would be the ice age. So this puts them within that range, which is that would be very difficult to square with the standard model because if we date the fossil by the preserved tissue, that would put it in range of the ice age, which would be very unlikely for a dinosaur to survive.
Yeah, yeah, exactly, because they're not nimble enough to survive an extreme condition like that. Um, yeah, but I'm sorry, man. I'm open to that option. It's not there's I don't know if any people from the audience maybe also, or if you've seen that on Joe Rogan. They had these archaeologists there, Graham, Graham something.
Graham Hancock.
Graham Hancock, exactly. And he basically said it may have been that in the past there was another civilization, and that civilization was destroyed by meteor swarms, something like that, right? So I think there's always a possibility that there's certain parts of our history are just more or less erased from our immediate ability to see them, right? We have to dig to find traces of maybe, or if we dig, we find traces of other civilization that we haven't known before, and then we would also have to rewrite at least the last 50,000 to 100,000 years, a little bit at least, right? But it's always possible that there were civilizations or that there's certain species in biology that just ended in a different way than we envisioned, just because we haven't seen the entire picture. It's entirely okay, and maybe I'm just maybe I'm too long removed from science. Maybe I haven't been in a lab for 10 years. Maybe I'm too open-minded, but I don't really, I don't really have a problem if a meteor didn't all of a sudden wipe all dinosaurs out. I don't know the evidence for that. Maybe there's great evidence for that. It seems to be the common situation, common model that archaeologists have settled on. But that doesn't mean that that it can't be that doesn't mean that it can't be shoved aside for a better model.
Well, right, right. So what I'm saying though is that, however, okay, I'm not saying that your version couldn't be the case, but what I am saying is though that this creates a serious problem for the established paradigm that I don't think it's going to want to deal with. I think what it's going to want to do is avoid dealing with this as long as possible.
I can absolutely see that because it sucks rewriting all your paradigms. But I mean, what has to be done has to be done. I
That's
mean,
science, right?
yeah, I mean, scientists are paid to do something, so we might as well just get active. I don't have a problem at all with that, but I know that there's it's so difficult in science to get new ideas through, and one point is if you find people become emotional against you, other scientists start really attacking you, then you're probably on the right path, right? It's just not
Yes.
fun on that path, but it requires on smaller new discoveries that are against entrenched paradigms takes 5 to 10 years until they hold true or until they're accepted, and then maybe in archaeology is it takes a little longer. I don't know, but yeah. To be honest, I came into this show with not thinking not much about dinosaurs being 68 million years old. I thought, yeah, of course, I guess that's the agreed on narrative. But now I don't have problem if they're not that old. I mean, it's not an earth. I mean, in a way it is, but it's not completely unlikely, completely improbable that these dinosaurs have been less than 68 million years old. And there's a lot of, like, when we say, like, the upper limit in how fast blood cells decay is 100,000 years or 50,000 years or whatnot. There's this huge insecurity, right? 50,000 years put it right in line with the ice age. But what if there's also like a meteor dryad swarm that didn't only wipe out humans but also a large chunk of the dinosaurs, right? There's always a possibility, and I'm not married to the number of 68 million years. It's just it is a large step, right? Going from 68 to 1 million years. But if you have the evidence, you have the evidence. I mean, you saw the pictures. You saw the pictures. You saw what these blood cells looked like. The blood cells, they look better intact than they look basically almost the same. They were they showed almost the same integrity as current ostrich blood cells and chicken blood cells that they had to compare. It's almost frivolous, you know. You think, wow, those are 100,000 years old. They just look as if they've been taken out of an ostrich. You
So
know.
basically now we have the new Florian Graham Hancock model where the dinosaurs were caused extinct at the Younger Dryas asteroid event.
Yeah.
There we go. No, that's great. So real quick, Middleton Walker, $9.99. Thank you, Middleton. Always appreciated. Um, but okay, the other thing too is to your point, Florian, right?
Mhm.
Imperator Ford says there's depictions of fossils in old Chinese and Greek vases. They're called mythological creatures. Oh, he's referring to vile crocodile. But we do have accounts, right, of dragons in China and Greek mythology. Now and even Pluto or Herodotus, the Greek. So it's possible, and I think this is where you're going. That well, maybe these dinosaurs were inspired the dragon legends.
Yeah, that could be. Um, you know, as I said, these pictures, they're not, we're not seeing pictures of demons. I mean, we can see them too, but we're not seeing pictures of demons with horns so much. We see pictures of mythological creatures, and they have to come from somewhere, right? If humanity thinks about mythological creatures, I get that these creatures can sort of like arise as part of a narrative, as part of like a story that's get told over generation to generation, and then it gets a lot of other stuff comes in there that wasn't in the original story, but still the form of the creature must come from somewhere, right? Otherwise, the mythological. I mean, why would mythological creatures look like dragon and giant snakes? Why? I mean, why would they don't? Why would they not look again like anything else? They must have some form of idea how these beings were shaped, and then they derive mythology from it, right? So a very good point. Very good. I think Imperator forced to remind us of that.
So yeah, it's possible that again, but from the point of view of however old we want to say that these soft tissues are, putting dinosaurs in the Cenozoic would definitely upend a lot of people's understandings of Earth history. That would be a big adjustment, to say the least. But it also then, the other problem though then with the dating, right, is that if that's the case, why is the rock dating so far off? Because this fossil was found in that rock layer. That creates another problem. Why would there be the divergence, right? The question is, why would they diverge at all? You wouldn't expect that.
That's a very good point, right? But so, I mean, maybe those ancient rocks were somehow pushed by more modern rocks over the more modern dinosaurs. I mean, I haven't seen what layers were in those sediments where they found the dinosaurs in. That would tell us more. But it doesn't mean just because you find fossils inside, or maybe not, just because you find these dinosaur remnants inside stones that are millions of years old, doesn't mean that they couldn't have been pushed there by younger rocks or younger sediment, right?
Well, I don't know what they would say about the rocks around the fossil. I don't know what that answer would be, but we do have more dinosaurs now than just the original T-Rex that the team discovered, and I think that might make it a little bit harder to say that all of those other fossil cases would meet that. So, but the other problem too, and this is something that I really would like an answer, and I don't think we have one. As Mary Schweitzer said, everybody would have thought you wouldn't find anything if you dissolved the fossil. The real question is, why would you do it? So when I watched the news break back in the day, when I was a kid, they said, "Oh well, Jack's an iconoclast. He's just a rebel," and they did talk about how it would be almost considered sacrilege to destroy a fossil because there's so few of them. But then, what? Clearly, they expected to find something. I don't think that you would do this if you didn't expect to find something.
In my life in the lab, I've never had any situation where I would just willy-nilly put some biological material under the microscope just to have a look. Let's see what I find. Maybe there's something. No, that's not how science works. You have a certain hypothesis. You observe certain things. You have a hypothesis, and then you do things to prove your hypothesis. And you destroy dinosaur bones because you had a hypothesis before, not because you just wanted to look whether something is there. I mean, this is a complete senseless. I mean, even if it's, especially if it's a surprising finding, why would Horner and Schweitzer just destroy their bones? You're absolutely right. That would be just senseless kamikaze. This is not how science works. Maybe archaeology works different, but no, biology doesn't work like that at all. And I'm sure physics and chemistry doesn't either.
And so, this is my unproven conspiracy theory that Jack and Mary are actually undercover creationists trying to discredit the age of dinosaurs. Because, and they actually admit the implications for creation. I'll reiterate it. Jack said to Mary, the creationists are going to love you. And she said, "It's your damn fossil, Jack." So that they knew, in private communications, what this could mean. And so, you could say, okay, do we take, I'm being somewhat tongue in cheek here, but I mean, why not? I don't know. It's like, um, it's very strange. Like, I don't really know why they would do this because, again, when asked by Bob Einhart to carbon 14 date the T Rex, it's like, no, we're not going to find anything. We're not going to do it. Okay, well, why dissolve the bone in acid? You're not going to find anything. It's all minerals. So it literally, and this was, by the way, a year after 2006 was Einhart. 2005 was the publication of Mary's discovery. The reasoning does not make sense. There's some sort of mental disconnect here with Jack that I just don't understand.
Maybe they weren't sure whether the dinosaur remnants they had were really that old. Maybe they thought maybe we find some biological tissue that's still preserved there. Maybe that was something they thought, or maybe somebody doubted that these dinosaurs were really those fossils they found. It's puzzling because nobody explains also why they might have done it in the first place, right? They only described an accidentally we broke the bone. So basically, what could have happened was when they took the bone, they broke it. So now I don't know how if fossilized, maybe fossils. This is just something they come up with, like out of thin cloth. So maybe fossils break along a certain way, right? And the way Schweitzer accidentally broke that bone, the femur, maybe that looked like this is no no fossil should break that way, right? This looks like it's biological tissue, not really completely stone. Or maybe they said this looks like it hasn't completely been transformed to stone. Let's look what else is there. Maybe that was what they did, or maybe when they broke the bone, they had some fragments flying around, and they took like one or two fragments and looked at them under the microscope, and then all of a sudden they saw, oh, this looks a little bit like biological material. Let's see what this really is, and they took some of these fragments and dissolved them, and then to see what they were left with. That's one plausible explanation. I think that can happen. Accidents happen in the lab all the
Yeah,
time, right?
yeah.
And so that's one way they saw they would have never looked at it, but they broke it, and then they saw, oh, okay, because you're curious as a scientist. You're curious. Okay, I broke it. How does it actually look from inside? Because nobody looks at bones from the inside, right? You broke it accidentally. Now you have a chance to really see what's in there. That would be my first step I do as a scientist. Someone smashes to the ground, say, I'm so sorry, but hey, look, what is actually inside? That could be one reason why they went, and it looked suspicious to them, so they just took some of these little crumbs and dissolved them and looked what was left. That's one explanation, I think.
It could be. The other thing I wanted to touch on was Mary Schweitzer herself comes up with some sort of mechanism, because one of the critiques I've been making here is what's the proposed mechanism for the preservation. And in 2014, she published a paper about experiments using iron-mediated cross-linking, where she, for a 2-year period, was able to preserve ostrich tissue. And I thought to myself, well, yeah, but that's not 80 million years. Like that, what you would need to do is see. This is what is going back to David Berlinski's critique of how biology is conducted. It's not quantified. The first thing you'd want to do is you'd want to come up with some proposed mechanism for preserving the tissue. Now, then what you'd want to do is you want to create a set of experiments where you expose soft tissue to a simulated version of whatever the predicted exposure would be, and then see what happens, or write a computer program that does that. So, like, for example, in industrial science, if you want to test corrosion, you can create a saline solution that creates an artificially high level of corrosion in a short amount of time, which represents 10, 20, 30 years. So, I mean, you can do this. I asked Grok, I said, has anybody done this? And it said no. And I'm like, okay, why? This should be like, with a groundbreaking discovery like that, this should be literally the next step.
Yeah, I mean,
You don't see
you're
it.
right, because they try to. So Mary Schweitzer showed not only that there were blood vessels she used she used ostrich as a species to compare to, right? What happens if you dissolve bone? Are you left with structures like that? And then so on, yes, okay, so they are likely then blood vessels indeed. But then, another experiment is actually, try to try to copy the phenotype that you observe by taking a live animal or like a dead animal, of course, and then just somehow find a way to with it to simulate the effects of erosion. Even if you can't do a million years, you would still see what happens to material when it starts eroding, right? And that already gives you some hint of if your model is at all plausible or whether there must be something else to it, right? If your blood vessels are all gone within like 10 years of your advanced speed simulation, right? Then then this makes less sense what you're seeing in terms of blood vessels. I mean, yeah, you can say that's not possible to simulate it, but you can simulate something, and I mean, Why wouldn't you just have a look? Nothing is perfect in
Yeah.
science, right? You make do with what you have.
Yeah. Okay. So I asked Grok this, and Grok says, why paleontology lags in accelerated testing? And this was specifically about Mary Schweitzer iron cross-linking. Several factors explain why paleontology has not yet prioritized accelerated erosion experiments like those you propose, subjecting iron-tested osseous tissue to conditions mimicking 100 million years. Well, no standard model. Unlike rust or drug stability, no established model exists to equate lab conditions to millions of years of fossilization. Protein degradation, kinetics, and bone microenvironments are poorly understood, complicating test design. Okay, well, this is actually a really big problem. If we don't even have a standard model for this, we're at the very, very beginnings of anything like science, because you have to have a model that you can use and then test it. If we don't have one yet, that's a problem. Complex variables, geological conditions vary widely across fossil sites. A single test might not capture this diversity. And oversimplified conditions could destroy tissue in ways not reflective of the fossils. Yeah, but point two is not really an objection. You got to at least do it once, and then work on the others. And then the third objection was the methodological challenge. These are all methodological. Even if tissues survive accelerated stress, proving they could last 100 million years requires fossil comparison, which is already available, reducing the incentive for complex simulations. Well, no, that's just begging the question. We have to do the test first to see if it's even possible. And there's funding problems. I get that. Philosophical differences. Oh yes, that's a big one. A lot of this just sounds like special pleading. I mean, if you were doing anything like this in the economy and not some dusty university paid for by the government, you would have to do this. And so it's like, well, why don't they? I mean, if you were going to do, for example, like with the double slit experiment for light, if you had that theory, you'd have to do an experiment to at least to prove it. Now, this is not necessarily going to prove it, but it would at least add weight to any sort of mechanism for preservation. And the fact is,
Yeah.
we don't have a mechanism for preservation, and nobody's even proposing a test whereby we could do that.
Yeah. Yeah. I mean, I don't probably archaeologists are probably working on that, but yeah, I mean it's not too long ago, or maybe it is 20 years ago, 2005. But the experiment with which she had basically used to show that this were blood vessels were comparisons with a bird, with bird blood vessels out of bones, right? That's a pretty crude measure, right? But you don't have anything better, so that's what you do, and it's actually a nice idea. And they had some mass spectrometry, they had some atomic analysis, and they had some biochemical and atomic analysis of the structures they were looking at. But still, yeah, maybe I mean I don't know. I mean, archaeology is also going into weird directions when they were like trying to show that some that some human fossils they found or human remains from people that died 10,000 years ago. They show, oh look, these were transgender humans or something like that. I
Yeah.
mean, I think paleontology and archaeology have had to deal with an onslaught of wokeness,
Yeah.
which has
Yes.
probably slowed down the science for a bit, at least. We hope that's over now, but that's also one reason why in certain scientific disciplines you don't do the productive experiments or don't develop productive experiments.
Well, yeah, and the other thing too is Mary Schweitzer was only one with her iron linking was only one form of preservation. Here's the thing: nobody's denying that these methods can add, can preserve the red blood cells or tissues a little bit longer than under normal conditions. The real question is, are any of these sufficient explanations for 68 to 100 million years? And the answer is no. In fact, we haven't even done the kinds of tests that you could even point to indicate that we have something like that. You'd have to have some sort of simulated exposure to. I mean, heck, let's just make it 500,000 years. If it can't even make it 500,000 years due to a simulated test, then we know it can't be 80 million. But
Right.
we won't even do that. And I'm like, okay, well, I think we're getting to the point where we were in an earlier show with one of our where we just talk about there's assumptions that are baked in. Okay, I know what it was. The preservation of adaptation of finch beaks, right? If it's environmentally conditioned, is it preserved if it leaves that environment or if the environment changes? And that's something that doesn't seem to be the case. So then, the assumption that these changes just roll over continually without actually trying to quantify, well, are they preserved from generation to generation? We're seeing this quantification issue here. And with paleontology, we just see so many just so stories,
Yeah.
and we see the refusal to engage in quantification. And the thing is, it's science. It's what you're supposed to do. I mean, what makes science science and not something else is that you can quantify it, you can measure it, you can do all this stuff. And if you refuse to do that, well, what are you doing?
Yeah. Yeah. If Imperator Forge said, the study of the past is driven by the bias of the present, and
That's a big part of it.
right, especially, I mean, physics, biology, or biochemistry, biomedical engineering. You can design lab experiments that show you the result immediately, right? Um, and I think also archaeology or paleontology is not really it doesn't have really an experimental mindset, right? They don't expect that they simply don't expect that there's ways that they could actually experimentally test the hypotheses. It just doesn't. It's not done, right? Because they have their set of tools that they use, and I can tell you one thing: scientists are very loath to use different tools from the ones that have always worked in their hands, because it's not easy to get an experiment to work. You want to have a reliable experiment to set up that has always worked in the past, and of course, that means you don't make many experiments and you don't really try to improve the tools that you have, right? That also plays a role.
Mhm. Well, the other thing that's suspicious about paleontology and Darwinian theory is they refuse to allow cross pollination with other scientific disciplines. So, for example, with intelligent design, when I would watch debates or discussions between Darwinists and intelligent design advocates, the Darwinists would commonly say, "Oh, but those people are engineers; they're not biologists." Okay, so that's supposed to be an objection? Like you talk about biological organisms as machines. That's the words that they use. So if they are biological machines, then an engineer has an awful lot to say about this. And then furthermore, the information theory, which we covered last week, they're going to cry foul and say, "Well, no, you can't apply information. You can't do that. That's just not how science works." So, for example, we have a field called biochemistry. So, chemistry does impact biology and then inform biology, right? You can't say no. We're not going to be informed by information theory. We're not going to be informed by engineering principles. We're just going to leave that out. Who gets to do that? I mean, you don't get to do that. If you were to study a biological organism today, you would start with the biology, of course, but you could dig a little bit deeper into the chemistry, and you could dig a little bit deeper into the physics, and all of that would create a full expression of the understanding of that biological system.
Yeah.
And you can't just compartmentalize this. And this is why I think a lot of this reminds me of the Soviet Union with Lysenko and all of this. It's like, well, we just can't do this. We can't talk about it. We're not going to compare. We're just gonna hermetically seal it off. And the question is, one, why? And then two, you can't do this in any other discipline in science.
Right.
So
yeah. I mean, there's a little bit of bio. I think that they use mathematics and physics to they apply that to biology at least a little bit, and I think they've mostly focused on genomic analysis, so basically to see which genes are there and how genes are structured and so on. But I mean, biophysics is not. I mean, they're not. Maybe if they say those are engineers, what they're really criticizing is that these people have too crude or too pragmatic of a worldview that they don't want to let them close to their precious biological specimen, right? There's always
yeah,
scientists sometimes find themselves better than sometimes scientists think they are better than mere engineers, right? So they are the ones who have a theoretical model, and the others are just some tradespeople that somehow got lost in science. I think
yeah,
that's one of the reasons.
yeah. So I think we've covered most of what I wanted to. But can you like what is your overall opinion on this shocking discovery and possible implications?
I think it's fascinating. I'm glad Schweitzer is doing more research on that. There are two possibilities. A, she's looking at artifacts all the time, or B, the dinosaurs are just not that old, right? I mean, it's not unheard of that you have like specimens that are evolutionary conserved and still alive, right? You have these fossilized fish that are still alive that live somewhere in the deep ocean,
The
and they're
coelacanth,
fine, right? Say again?
yeah, the coelacanth.
Yeah, I mean, it's not. Why isn't it possible that some dinosaurs survived until 100,000 years ago, until they finally withered away for good? But if it's not an artifact, then I would be really interested in. I mean, Horner should just take some speck of the bone and send it to carbon dating. It's an easy
Yeah,
thing. It's a
yeah,
service, I believe. They probably can just send it off and get the result in 1 week, or maybe even faster. So and with that quick flick of a wrist, basically, he can settle the entire thing, put it to rest.
he could. This was somewhat tongue in cheek, but Seamus O'Blaine says evidence for time travel or temporal dislocation. the the younger dinosaur fossil being found in the older rock.
Yep.
That's funny. Let's see. Yeah, well, like I said, I'll reiterate my three what I think are three things that follow from this. One is we have the first crisis, which is the actual preservation of this tissue at all, which Schweitzer admitted one would not have expected based on the standard paradigm. Two, the dating difference. Why is the rock so much older than what it looks like this tissue is? And then thirdly, if this tissue does indicate the animal was much younger, like around the time of the last ice age, that certainly would force a lot of people to reconsider and rediscuss the whole life history of Earth. Would it be possible? Sure, but it would be a real challenge, and I think people might decide to go some other way instead of that. I actually think a lot of these problems are going to indicate that intelligent design becomes the new paradigm. I think creationism makes too many specific claims that are going to be harder for people to swallow. But intelligent design, I think, could become the new paradigm, and that it doesn't stand or fall on the age of a T-Rex fossil. Some intelligent design people might believe in a younger animal, some might not. It doesn't really matter. So I think intelligent design might become the new paradigm because of all this.
Mhm.
I'll ask you, what do you think about that as an idea?
Yeah, I mean, if you're looking at science, then the more specific you can be, the better. But I think if you really want to understand everything, you should look at the whole situation, right? But for scientific analysis itself, it's better if you have something that is more specific to a certain area of the science you're looking at.
All right. Well, thank you, Florian. It's always a pleasure having you on. This is Todd Lewis of the Praise of Fly podcast signing off.